La siguiente sesión es en inglés, so I welcome Dr. Yassin Suilmi. He studied his bachelor's and master's degree as well as his PhD at the Mohammed V University in Morocco. He worked as a researcher and assistant at Harvard University, focused on bioinformatics. Currently, he's a postdoc at the Australian Centre for Asian DNA in the University of Adelaide. Thank you very much, Dr. Yassin, for accepting the invitation, and we're very much looking forward to your talk. Please have the floor.
Thank you very much. Thank you very much for inviting me, and it's a pleasure to speak at your event today. I think, so as you said, as Alejandra said, I work at the University of Adelaide at the Australian Centre for Ancient DNA, but at the same time, I have an affiliate position at the Australian National University, where I work at the National Centre for Indigenous Genomics. This is a recent transition, so we do a lot of work, genomic work with Indigenous Australians on multiple fronts, so mostly focused on generating resources for to improve medical care in Indigenous Australians. So I'm going to answer the first question probably everybody is wondering is, where the hell is Adelaide? So this is the map of Australia, and Adelaide is down here, and it is based in Ghana country, which are the traditional custodians and owners of the Adelaide Plains, where the University of Adelaide and the City of Adelaide is located. So I would like to acknowledge their ownership, their elders, past, present, and emerging. And so it's a really beautiful place, and that's probably one of the only ways I can endure the 24-hour flight from my home country, Morocco, down here, maybe once or twice a year. So getting back into the presentation and the spirit of why, the reason why I was invited here. So a little bit of a step back. So if you look at human history, throughout we are migrants. We are a species of migrants, and since very early on, as you can see, we've been moving around for thousands and thousands of years. And as we were doing that, our ancestors have occupied varied types of environments, from the savanna of Eastern Africa, where we think our species have emerged, to deserts, forests, tropical environments, high altitudes, and extremely cold and arid environments. And while we were doing that, we adapted to these environments, but one of the things we have adapted to are pathogens. So as we were migrating, we were exposed to pathogens either from cousin groups, think about it like hominid groups and Neanderthals and Denisovans, as we met and mingled with those cousins, but also sometimes every now and then you have the odd pathogen that we get exposed to from our exposure to other animals, either through hunting or sometimes even foraging, or being exposed to bacteria or other pathogens from soil and other trees and other things that we get exposed to, and the occasional zoonotic infection, like many of today's speakers have been talking about. And so what does adaptation to a pathogen look like? It's pretty similar to any stimulus. So you could swap a pathogen here with any other environmental stimulus that is different, that we're not used to being exposed to. And think about the different colors representing the population here as a simple locus in the genome. So maybe a gene or a variant. And so as we're exposed to this new pressure here, a pathogen in our case, over time, the genetic profile at that particular locus of the entire population sort of gets homogenized and looks very homogeneous, and one type, one allele gets overrepresented over the other alleles. So in this case, the orange or the coral color allele is overrepresented when compared to the blue and the white alleles. And this obviously happens over a long period of time. You need several generations, so at least probably 200 or 300 generations to be able to see this effect. And so the way we detect this, and this is a very simplified way to explain this, is think about the red graph here is sort of your background level of diversity in your genome. So if you take each one of, if we take, like, there's probably nearly 200 people on this call, on this conference call. If we take 20 people randomly from here, and we measure the genetic diversity at each position of the genome, and what I mean by genetic diversity is the number of different alleles at each locus, and we plot that, you have this background variation, and it fluctuates a little bit because there's a bit of randomness there. But however, if you have a selection on a particular locus, you can see that the genetic diversity around on the flanks of that locus is similar to the background, but then it drops at the locus and then it increases again. And when you look at it from a variation point of view, here what I'm plotting, like, think about it as sections of chromosomes aligned to each other. And we don't care when we are doing analysis like this, at least in many cases, about where the chromosome's coming from. So this chromosome can be coming from, and this one can be coming from different individuals. And you look at this level of diversity that we have here, and what we have is that these red variants are neutral, so they don't affect the trait that is selected. And the blue allele is the beneficial allele for that to respond to that trait. So in our specific example, that could be better survival, better resistance, or better detection for a viral infection. And so once you, over time, you can see that this allele, that you have this beneficial allele almost fixed in this case, so it's 100% frequency. And then on the other flank, you just have background level of diversity again. And so we applied this kind of principle. We asked ourselves the question, well, what if we do that and look for adaptation to ancient forms of coronaviruses? And what can we learn? And what can that teach us for the current pandemic? And so we published this paper last year, and we applied the same principle to scan for signatures of selection in the 1,000 genome dataset, which is represent 26 different groups around the world and has about a little bit over 2,500 samples. And so one way to distinguish the signatures of selection of the different signatures of selection that we detect around the genome, because we agnostically scanned all 26 populations and we scanned their entire genomes. However, we have the benefit here that from the literature, we know what coronaviruses, including SARS-CoV-2, which proteins in the human genome the virus interacts with. And so when we contrasted the signatures of selection in the rest of the genome versus on the viral interacting proteins, or the SARS-CoV-2 viral interacting proteins, we found that basically we have more viral interacting proteins and their selection, or exhibiting signatures of selection, than expected by chance. So obviously we know the number of genes in the genome. We know the number of viral interacting proteins that interact with specifically with SARS-CoV-2. And then with using that proportion, we can have an expected value of how many of those are will be coming under selection if this signal was random. However, we have a very significant number of viral interacting proteins that are under selection that expected by chance. And so there are multiple ways you can do this. The way we chose to do it in this study is we use ranks. It makes the statistics very simple. So the way this works is you take each one of the SARS-CoV-2 viral interacting proteins and you give them a rank based on the score that they were assigned by the selection statistic. And we used 3 different statistics, and they all gave the same results, more or less. And then you rank all your genome, all the genes in your genome that are under selection. And then you split them into 2 groups. There is the SARS-CoV-2 viral interacting proteins, and then there's the rest of the genome. And then we plot the ranks. And so, for example, the way you read these plots is that you say, I have 20 times more SARS-CoV-2 viral interacting proteins with a rank of 10 or less. And that's how you read that. And then again, same thing. I have 20 times more viral interacting proteins than that are ranked 20 or less, etc. And so this tells you that essentially, using this NSL statistic, that there is a very high enrichment in viral SARS-CoV-2 viral interacting proteins that are under selection in each one of these respective populations. So you can see that we have the Dai Chinese, we have Han Chinese, South China, Japan, and Vietnam. And then once you get to other populations outside of that geographic area of Southeast Asia, you have the signal is very faint, which is suggesting that there was a significant selection on SARS-CoV-2 viral interacting proteins in this region. So the region of China, South China, Vietnam, and Japan. And so we wanted to take this a step further, and we use this method called iSafe, that it takes a sweep region. So as you know, DNA in humans and other mammals is linked. So when you have a sweep or a selection against one variant, generally speaking, it will drag along with it to high frequency all the variants that are linked, or all that segment of DNA that is linked to it that hasn't been broken by recombination. And so there's this method. Essentially, this method will basically try to identify which one of those linked variants is under selection. And the way it does that is that it actually introduces artificially variation from other populations where selection is not inexistent or weak. And it tells you which one is the likely favored mutation in this case. And so after we've done that, we identified the putatively driver mutations. We used a secondary method called relate that actually takes the locus and builds these ancestral recombination graphs, which essentially think about them as, instead of building a phylogenetic tree for the entire genome, you break your genome into small chunks and you build a tree for each one of those windows, and then you slide along a little bit, and then you slide along a little bit, and these are overlapping windows. And by rebuilding these trees, you can do a few things. Particularly if you introduce ancient DNA samples, you can actually calibrate your mutation rate for that sample. And by calibrating your mutation rate, you can actually, for each one of these little windows, you can create, it's essentially a coalescence tree, or you can estimate these split times. And subsequently, if you take a specific variant, for example, the blue variant in this tree as an example, it can tell you when that allele emerged. It can also tell you, because you have the data from multiple different populations, it can tell you how the frequency of that variant changed over time. So it will tell you the accumulation on the frequency curve. And so combining these two methods, what we have is we have an allele, we have an estimated time of emergence of each one of these alleles, and we have also the time of when the allele frequency started increasing sharply for each one of the selected putatively selected variants that we identified for each of the selected loci in the genome. And so when you plot the entire genome, you get this blue curve. And when you plot the coronavirus's viral interacting proteins, you have this peak between 700 that started between 770 generations ago and 970 generations ago. And when you translate that, you multiply it essentially by the average generation times for humans, which is 29 years. It gives you around 20,000 years ago. And so this is essentially, when you plot that for a specific population, you can see, if you zoom in here, you can see that the inflection point and that exponential phase of allele frequency increase is around 900 generations ago, and that's about 20,000 years. And so essentially what we have identified here is a significant level of selection on coronaviruses in the viral interacting proteins. And this selection was very sharp and very strong, and happened about 20,000 years ago when you aggregate the signal. And so this is essentially the first, it's circumstantial evidence. We don't have physical evidence. We don't have ancient data or remains with viral proteins in them. But this is circumstantial evidence of the first large-scale pandemic in humans, or viral pandemic in humans that we know of. And so we wanted to get an idea about what are the biological processes that these viral interacting proteins and their selection are implicated in. So we did the gene ontology enrichment on this, and very well you can see that it impacts genes that help in the nuclear division, translational elongation. So essentially, a lot of biological processes that are involved in cell division. And that is essentially, that makes sense, because when the virus invades the host, they don't have their own tools to multiply. So they hijack the host cells to multiply themselves. And that's why they promote nuclear division processes and translation, etc. All these processes that help them create multiple copies of themselves, which was a very neat result. And the other thing that we did is we essentially, to identify these regulatory variants, we actually, so we know that there is a genome-wide association study that was conducted on SARS-CoV-2, but also on other SARS patients back in the past. And you have these eQTLs. So these are expression QTLs, and we repeated the same thing with GWAS variants as well, is that we calculated the distance between the putative selected variant and each one of these selected SARS-CoV-2 viral interacting proteins, and the nearest EQTL. And we did the permutation test. That's what's depicted right here. And then what we found that these selected viral interacting proteins are actually significantly overexpressed in the lung, which makes a lot of sense because that's the primary tissue infected and affected by a SARS respiratory infection. Ignore the testes here because it's in expression data. Every single gene you can think of, more or less, is overexpressed in testes. Stomach is also secondary tissue that is affected by a SARS infection. You have the heart as well and the adipose tissue, which all makes sense here.
1 minute, please.
Yep. The other thing we did is we actually asked ourselves the question, well, right here, we are detecting signatures of selection to ancient forms of SARS-CoV, of coronaviruses. Well, how does this result translate to the SARS-CoV-2 virus that we have today? And so what we did, we actually took all 42 selected coronaviruses, viral interacting proteins that we detected in this study, and we actually did a binding cell-free expression protein. So we binded the SARS-CoV-2 specific proteins with their alleged targets in the human genome, and we found that 35 out of the 42 selected coronaviruses, VIPs, actually do bind to SARS-CoV-2 proteins, which is amazing. It's 80% binding success rate, which means that these results that we found and these 42 selected genes from 20,000 years ago are relevant to the current pandemic. And to extend that, we took the 42 selected genes and we actually looked in drug bank to see how many of these genes are actually targeted by drugs either used to treat COVID-19 infection or are being experimented on or trialed to treat COVID-19. I didn't update these results since late 2020, I'll be honest, but there are several drugs that have been used or are still being trialed, like ribavirin, for example, to treat SARS-CoV, which is amazing because it's actually suggesting that we can use selection on viruses from 20,000 years ago to identify putative drug targets today, and by extension, we could identify candidate drugs that could be repurposed to treat viral epidemics. And so I'm very excited about this result because it can potentially help us speed up the process of narrowing down the potential drugs that could be repurposed for the next pandemic if we ever get there. So just to summarize, we essentially found a significantly high level of selection on coronaviruses, viral interacting proteins. We timed this selection to around 20,000 years ago. We found that these genes are help promote and or prevent the viral replication, and we find that these genes are overly expressed in the lungs, as well as we identified candidates for drug repurposing as well to treat this viral epidemic. Just to conclude very quickly, please get in touch with me if you're looking to do a PhD or a master by research. I have several projects in the evolutionary space, but also in the indigenous medical space with an evolutionary twist. And thank you very much for listening. Thank you very much for inviting me today.
Shukran. So now we're going to have some questions from the audience. So the first question is from, well, we have a couple of questions from Federico Sanchez. So do your results of a previous event of exposure to coronavirus in East Asians translate to a better outcome of SARS-CoV-2 infection in East Asia versus the rest of the world for the current pandemic?
That's an excellent question that keeps coming back all the time. Probably, but our suspicion is that the effect of, or the protective effect of this polygenic signal that we found is probably very small. And I think it makes, so when compared to public health policies, prevention policies, I don't think the protective effect is that important.
Sorry, second question from Federico is, are the regions of the genome that detect ancient coronavirus specific to coronavirus, or could they be a signal that was due to another pathogen which is detected by the same human proteins in East Asians, giving the sign?
So, yeah, so the signal is specific to coronaviruses.
Sorry, your microphone muted.
Muted on its own. Sorry. So I was saying that the signal is specific to coronaviruses as a family of viruses, not to SARS-CoV-2. But they're not shared. We made sure that these viral interacting proteins are not shared with other pathogens. So as a control, if you read the paper in the supplementary, we looked at also at other viruses too.
Okay, we have comments of Red Talk. It's very interesting, the subject related also to the drugs treatment. I would also like to add that I wasn't like really sold on the idea until you showed these GTX and EQTL results. That was very impressive. And then the part of the proteins and the drug treatment, that I think these clinical trials on these drugs, I think they look very impressive. So great work, and thank you very much for joining us in this event. It was a very nice talk.
My pleasure, my pleasure. Thank you very much for having me today.
Thank you. Also, thanks to Federico Sanchez, who was very kind to contact you and invited you to this event. Para todos los demás, mañana continuamos de nuevo a la 1, así que los esperamos. Tenemos muchas pláticas igualmente interesantes. So thanks again, everybody, and have a nice evening. See you tomorrow again at 1.
Gracias.
Shukran. Bye.